Everybody recognizes aging. And my disclosures obviously are that I'm an advisor for one of the companies that's exhibiting here, Lifelength, and that's my only disclosure. There's a lot of promising new technologies in aging research, and this is a list of some of the areas. They're certainly not all inclusive. But what I want to point out is a lot of the things that many of the people here at this conference are involved with: anti-inflammatory agents, antioxidant supplements, various things that maybe mimic caloric restriction, things that alter energy, stem cell therapies, which I know many people are interested in at this meeting, all basically have an impact on the bottom one, which is telomere extension or cell rejuvenation. And what I hope to do is give you basically an overview of the science behind this new evolving field, and end up by talking about some of the tests that are available for measuring your telomeres. So first of all, let's just start off by saying, what is aging? Aging is associated with the gradual decline in performance and reserve capacity of organ systems. Everybody knows that. But maybe everybody doesn't realize that aging of organ systems is often associated with a decrease in the number and or function of cells. And we know that old cells, not just dead cells, but old cells do not maintain and repair tissues as well as young cells, leading to a decrease overall vitality. So we need to understand what aging is at the cellular level. And this sort of area became more well understood starting in about the 1930s, when Dr. Muller and Dr. McClintock both independently wound up studying these special structures at the ends of our chromosomes. And so without going into detail, the question that you should be asking yourselves are, why should I care about telomeres, right? Well, chromosomes, we get 23 from your mother and 23 from your father. And so you have 46 chromosomes, and each chromosome has an end. And so you have 92 ends, and the ends of our chromosomes are capped by these structures called telomeres. And the telomeres are there to prevent our cells from being recognized as damaged DNA, and therefore they sit there and protect our ends. What we've learned over the years is these things we call telomeres are actually repetitive elements. They don't encode for genes. They're just tracks of this monotonous sequence, TT, A, G, G, G. And you have thousands of these repeats at the end. So that's a good thing, because there's a problem called the end replication problem. And Jim Watson, the Nobel laureate, described that in 1972, the way DNA replicates. Every time a cell replicates, one of the strands, the lagging strand, is going to lose a little bit of the ends of those chromosomes. That's just the nature of the way DNA replicates. And there's also things like oxidative damage and other things that can also erode on your telomeres. And it was proposed that senescence, or aging, occurs when a few telomeres become short. So the way to think about telomeres are like the plastic tips on your shoelaces. When you're young and healthy, or you have a young, healthy, or young, quiescent, non-dividing stem cell, you have your cells capped by these little plastic tips, auglets, that are on the ends of shoelaces. When you're old and you lose this little plastic tip, your cells basically get a signal to stop growing and to shut down. So the question is, what's the evidence that telomeres have anything to do with senescence or growth arrest of cells as we age? In the 1950s, Leonard Hayflick showed that if you passage cells in culture, that eventually they stop growing. And when they stop growing, as you see over here on the right side, they get large and they actually don't do very well. And then in the 1990, it was shown these things, which we can measure here by one technique called TRF, our telomere restriction fragment, is a fairly cumbersome technique, but it gives you a general trend of the telomeres getting shorter with increased doublings. At the same time, a group in England had showed that if you looked at tissues from young and old people, that telomeres were shorter, even in dividing cells, such as the colonic mucosa blood cells, compared to fetal tissue or reproductive tissue. So, again, both of these cases, in cells and culture, as well as in vivo in people, telomeres shorten with increased age and passage, and these are both correlations. They certainly do not prove that short telomeres cause aging. So what are some of the things we're learning about short telomeres? Well, if you have short telomeres and your cells stop dividing, you lose tissue renewal capacity. You have a failure of stem cell niches. They can't maintain the stem cell population. You have problems with mitochondrial function, metabolic dysfunction. And the worst part of it is this next to last one, senescence-associated secretory phenotype. It's been shown that SAS actually results in senescent cells secreting a different constellation of proteins that may, in fact, make you at increased risk for the development of cancer and genomic instability. So the aging is basically a two-edged sword. You know, when we're young, we have almost no risk of cancer except for a few pediatric cancers. But reaching about age 40, the incidence of cancer starts going up enormously. So as part of getting old, you get shorter telomeres, and as you get older, you increase your risk of cancer. So our belief is that this shortening of telomeres, in fact, may have evolved as an anti-cancer protection mechanism. This cartoon basically says, today we know you need 7 or 8 mutations in an individual cell to go from normal to cancer. And what this says is, in order to accumulate all that, your cells are going to have to divide a lot of times, and they may, in fact, reach a senescence brick wall prior to becoming an immortalized cancer cell. What we also know is if you look at pre-malignant, precancerous lesions, that almost all of these, ductal carcinoma in situ, PIN lesions for prostate cancer, all have critically short telomeres. So the problem is, as we get older, our telomeres get shorter, and therefore, is that a protection mechanism, or is it, in fact, a genomic instability mechanism that might, in fact, increase our risk of developing cancer? And the way I look at this is that in the absence of other changes, having short telomeres may not necessarily be bad, but in combination with other alterations, the 7 or other mutations or epigenetic changes that need to occur for you to get cancer, could contribute to cancer. And the reason we think that is because almost all cancer cells are immortal, so they have to engage a mechanism for stabilizing those telomeres. Otherwise, your cells couldn't grow. And it's one of the hallmarks of cancer. This is sort of the famous Hanahan and Weinberg results that said enabling replicative immortality is one of the things that almost all cancer cells have to do. And the way they do that is by upregulating this enzyme, which we call telomerase. So telomerase, as you see here in this little moving pictures here, is a molecular motor that adds new DNA onto the ends of telomeres. And normal cells don't have this activity. Cancer cells have this activity. Normal cells don't have the messenger RNA for telomerase, and cancer cells do. So that's actually an explanation for why almost all cancers in humans engage this pathway. Obviously, this was a significant finding, and in 2009, Dr. Blackburn, Dr. Shostak, and Dr. Greider, illustrated here, received the Nobel Prize for the prediction and discovery of telomerase. So now that we've discovered it, what do we need to know about this? Well, it would be nice in cancer if we could inhibit it, and as part of aging, maybe slow it down or activate it transiently. Now, the interesting thing is that individuals with malignant tumors but without telomerase actually have better outcomes. So here's a group of pediatric patients. Their telomeres are very short, and they can't turn on telomerase. So if they can't turn on telomerase, their outcomes are really quite outstanding. So that's really suggests, as shown down here, that inhibition of telomerase may be a very potent anti-cancer approach. There's lots of ways that people are doing that. They're in phase 2 and phase 3 clinical trials. I don't have time in this short meeting to talk about all the exciting clinical trials in trying to inhibit telomerase as part of anti-cancer therapy. One of the areas that we've been working on is this drug called imetelstat. It's a direct telomerase enzyme inhibitor. It's not really critical that we go through all the details of what it is. The bottom line is it inhibits telomerase and telomeres shorten. And we have shown, and other people have shown, that if you take cancer cells with short telomeres, that we can inhibit their growth in cells and in animals, etc. And this has led to a series of advanced clinical trials. And the trial that just ended very recently enrolled 116 patients that received their standard care chemotherapy, and then in the maintenance setting either received imetelstat, nothing, or imetelstat plus an angiogenesis inhibitor, bevacizumab. The exciting outcome of this is the following, that these people that were on the imetelstat, the telomerase inhibitor arm, actually showed a slight but statistically significant increased median survival and a non-statistically significant overall survival. What I thought was even more impressive on these studies, as shown on the next slide, which is if you look at the progression-free survival or overall survival, and you look at individuals with short versus long telomeres, those that were on imetelstat that had the shortest telomeres actually had the better outcomes because it takes less time for the inhibitors to drive the cells into apoptotic cell death. So cancer patients with short telomeres responded better to these telomerase inhibitors. So what this says is that in the future we'll be picking, in this case, lung cancer, but this is probably true for all other types of cancers, with the shortest telomeres that have rapid progress of disease, and those are the ones that are going to benefit the most from telomerase inhibition therapy. And while not statistically significant, patients on the imetelstat arm showed an overall survival trend, and it suggests that patients with short telomeres may be an enrichment biomarker. So those of you that are oncologists may want to think about how we will use this in the future. It may be that every patient, before they get their chemotherapy, will have a biopsy. We'll measure their telomeres, and those that have short telomeres may be more appropriate for being put on an anti-telomerase therapy. So this brings us to the two phases of telomerase. What I've been talking about just this last few minutes is about the cancer-permissive part. When telomerase comes on, it actually results in a bad outcome, and then we have to inhibit it. But we have telomerase on, what I call the Mr. Hyde component of the Jekyll and Mr. Hyde, the aging cell rejuvenation component, because we know that telomerase is on in certain reproductive tissues, and we know it's on in certain proliferating stem cells. So we showed, as we heard from the introduction, that if you introduce telomerase into normal telomerase silent cells, that we not only turn on telomerase, we elongate telomeres, and cells functionally become immortal. Now, what you should be asking yourself is, if you put telomerase into normal cells, do they become cancer cells? Because that's what I just told you, that almost everybody with cancer has telomerase. And the answer is no, because these cells don't have the other 6 or 7 mutations. These are normal cells. So if you manipulate normal cells, there's absolutely no increased risk. And this slide just shows they don't make tumors in mice. They are karyotypically the normal chromosome counts and stuff like that, which is really different from cancer. The only thing that these cells do, that they basically can grow indefinitely, and that's what all normal cells can do, except living stem cells in our body can't grow forever. That even though they can grow longer, they do eventually also go down, and that probably was also to prevent people from getting cancer at an early age. So now we have evidence of telomere shortening in a whole series of human genetics and chronic diseases, and this table just lists chronic wounds, myeloproliferative disorder. All of these have extraordinarily short telomeres. The exciting one, and I'm going to talk about two real quickly here, dyskeratosis congenita is due to basic anaplastic, aplastic anemia. These people come in with all sorts of problems. And the other one is idiopathic pulmonary fibrosis. It used to be an unknown origin, and we've now discovered that some types of pulmonary fibrosis are due to mutations in either one of the two components of telomerase. And this is what the structure sort of looks like, and this is just showing you a cartoon. There is now a whole series of diseases which we're calling telomeropathies that are due to progressively, prematurely turning on short telomere, having short telomeres. So here's a case of a family with pulmonary fibrosis. You can see this lady right here. It's kind of hard to see from the back. Has very short telomeres. She's somewhere down here. And she has a daughter, 26 years old, that's somewhere right up here. But this is a sort of the standard loss in telomeres with age that we see. That when you're young, you have 8 to 10 kb of telomeres, and as you get old, they go down to 4 or 5 kb. And that happens in all tissues. And as you can see, all these people that have this type of mutation, with or without the disease, all fall below this graph, the average line. So while there's some heterogeneity in normal individuals, maybe due to genetics or environment, as we'll talk about in a minute, people that have a genetic disease where telomeres can't be maintained all have shorter telomeres than age-matched controls. And here's another example from dyskeratosis congenita showing that these people with a very early onset of their disease have extremely short telomeres. So this really shows short telomeres does correlate with disease in many times. And the way to think about this is, in these diseases, we get premature telomere shortening, leading to a premature growth arrest, cell senescence, progenitor stem cell depletion, and eventually clinical disease. And this really says that if you don't have a full complement of telomerase, you're in trouble. And it really says that we don't have a lot of extra telomerase in our bodies. And it was probably evolved to be very carefully regulated so you don't have too much or too little. And this is another way to look at it, that normally what happens throughout life is your telomere lengths shorten. And when you hit 70 or 80, you start getting certain age-associated diseases. These people with telomeropathies wind up having these much earlier in life. So tell me something else about telomeres and disease. Well, it turns out there's been thousands of studies, and a lot of this stuff has been published in the absolute top journals. Reduced telomere lengths are found in patients with cardiovascular risk factors such as atherosclerosis, hypertension, obesity, diabetes, smoking, physical inactivity, stress, and other types of chronic infection. So anything that's affecting your body is also affecting your telomeres. And shorter telomeres have been associated with an increased incidence of disease and poor survival. And just the opposite is found. People that have healthy lifestyles actually have longer telomeres. And so we're going to talk about a few of those in the last part of my talk. The idea is that normal people, again, reach this sort of age-associated increased risk of diseases using their 70s or 80s. However, a lot of you that have your own businesses, reporters, people that are stressed out, may in fact wind up having these occurring a little bit earlier. And we just as a society need to start dealing with biomarkers that can tell us if we are affecting ourselves. How do you measure stress? That's a good question, right? We know how to measure cholesterol, we know how to measure your blood pressure, but how do you measure stress? And this may in fact be a very potent biomarker for measuring increased stress. And we know there's a lot of things out there from Nobel Prize winners, etc., that things like vitamin C and vitamin D and all these and omega-3, a lot of these things can reduce oxidative stress, can reduce inflammation, and theoretically they could have an improved role on telomeres. But we have to know that for sure. You can't just say it. You have to do the double-blind clinical trials to show that these things actually have these effects. And so far, not many people have actually done those clinical trials. The part of telomerase that I think is very exciting is the possibility that instead of letting your telomeres get so critically short that you wind up getting senescent cells or an increased risk of cancer, is what if we were to give somebody transiently something that would activate or slow down the rate of telomere shortening? So that's the kind of idea that we're thinking about. And as you know, there are drugs out there now that have been isolated from plants that are claimed to be able to elongate telomeres. That still remains uncertain. There are trials being done as we speak to prove if these things actually do work, in which case that would be a very important advance. But so far we don't have those data in. So what would be the effect, you should be asking yourself, of turning telomerase on? Well, the good news is it would slow the rate of telomere loss. That's what we want. It would improve, hopefully, our immune cell structure and function. It would prevent or slow down the rate of genomic instability that's part of getting cancer. It would activate certain renewal pathways and potentially increase repair, resistance to stress-induced cell death, etc. The worry is, and of course it is a legitimate worry, that if you take the ticking clock off and now cells don't senesce anymore, they don't age, you are increasing the number of doublings from, let's say, 100 to thousands, and therefore, just by chance alone, you're going to increase the chance of mutations occurring and possibly increasing the risk of cancer. So that's the pros and the cons. So the way to really think about this, again, is using the same cartoon, that if you're a normal person and your organs and stem cells are all doing what they should, theoretically, when we hit 70 or 80, we should be starting to get some onset of some mild, if not serious, age-associated risk. When we reach 90 and 100, if we reduce this stress, better nutrition, better exercise, reduced inflammation, potentially telomerase activators, maybe we will be able to compress the morbidities associated with increased age into a much smaller period of time. So we come to the sort of last part of this lecture, which is, are telomere tests ready for prime time? And I think that's a completely legitimate question. There's a lot of people and companies that are out there saying, we can measure your test. And what I'd like to do is just go through the science of testing and what I think are good and bad things that you want to find out. So what do you need to know if you want to determine your biological age? You know, a lot of people here look a lot younger than their chronological age, but what do you really want to know? And why do you really want to know your biological age? That's a fair question, because if you know that you've got something wrong and your chronological age is actually less than your biological age, that means you've got a problem, and we need to deal with that. And that a lot of you have practices, and there are ways we can reduce that. So it's an excellent biomarker. If your biological age is greater than your chronological age, what does this mean, and what can it be done to slow down or reverse this? What is the difference between average telomere length and short telomeres, and why is this important? What is the standard normal telomere length by age? What is the best and most accurate test? And what cell type should you test? I think these are the kinds of questions those of you that are thinking about incorporating telomere testing into your practice should be thinking about. And these are legitimate questions, and we have answers to many of them, but not necessarily all. So this is a really difficult slide to see, but I can simplify it by skipping to the next slide. What you really want to know is if the test is robust. There are some tests like Q-fish telemapping, which are very low throughput, which means that it's going to take a long time to do it. It's going to be very, very expensive. There's biological laboratory things called Stella that are also very difficult to do, and single sampling. So what you want is something where you could do multiple sampling, and preferably one that will give you how many short telomeres you have. And so we only run into a couple that can give you some of that information, and I will be glad to show you the data on that. So again, a Nobel laureate, Dr. Carol Greider from Johns Hopkins, published a few years back that it's the shortest telomeres, not average telomere length, that's critical for cell viability and chromosome stability. So having information about the average telomere length may or may not give you very much information. You really want to know what percentage of your cells have critically short telomeres, because it's only when cells have short telomeres that cells undergo this senescence or growth arrest. So that's actually quite important in your consideration in thinking about testing. The test that is used for measuring the shortest telomeres involves a technique called fluorescence and Situ hybridization, or FISH, for measuring it. Basically, if you can see this little cartoon of a chromosome, this end of this chromosome has two bright spots, and this end of this same chromosome has two weak spots. And so you can actually set up very standard procedures for measuring how many short and long telomeres you have. And this is just a machinery of one example of how you would do it. You would collect the blood sample. It would be shipped to a standard laboratory, and then it would be processed under automated, mostly conditions, by high-throughput quantitative FISH, put into a very expensive machine, and then it images your cells. And this is from peripheral blood. And after they do that, it gives you quantitative readouts, and then the patient and the physicians get back data, which is always sent directly to the physician. And then in case the patient wants to have a consultation to understand what this result means. Now, the reason why the percent of short telomeres is important is because this is what normal telomere shortening looks like. Lots of heterogeneity, right? But it's all pretty close. So the difference between a 70-year-old out here and a 40-year-old is really not that much in telomere length. So average can give you a trend, but look at the scatter of short telomeres as you get older and older. There's a lot more scatter, and this is where we're seeing differences in health outcomes of patients. Those patients that are 70 that have a lot of short telomeres, like these people down here, are generally have something going on. These people out here, well, I can't promise you how long you're going to live, but that's where you'd like your telomeres to be when you're 70 years old, because that means most likely your stem cells are doing well and you're doing well. So the percent of short telomeres detects more differences between individuals than average length. The percent of short telomeres shows higher scattering as we age, which is good, and because you can separate out patients better that way. and the percent of short telomeres are likely to reflect both genetic inherited differences and lifestyle choices or environment. I just quickly mentioned that you can look at this in tissues. Remember, I mentioned most premalignant tumors have short telomeres. Here's a patient that has a preneoplastic lesion in the prostate. You may not be able to see that, but there's these green cells here with little red dots in them. And if you were able, in the front row, you could see there's lots of red dots here in these luminal epithelial cells and almost none in the pin lesion. So what this basically says, again, if you're not looking at the shortest telomeres by looking, there's no way to know anything, because all these long telomeres would just sort of blend in and you'd come up with an average, and it wouldn't be very useful. So now the question is, what type of cell should we use? So there's some individuals that have tried saliva, including Nobel laureates, and the trouble is, why is that good? Well, saliva is easy to obtain, and testing can really be much less expensive. However, saliva has a lot of problems. The problem is they contain few living cells, so you have statistical problems of getting accurate readouts. Poor quality. The DNA in your mouth is, unfortunately, fragments very easy. These are cells that are dying primarily. And you also are contaminated with bacteria in this sort of things. And, in fact, the worst part about it is there's almost no published data comparing results to other tests. Only average telomere length is obtained. So almost all the published work, everybody that's legitimate in the field, has basically been using peripheral blood, because that's an easy thing to do. People get their blood taken all the time, and it's just a simple thing of taking an extra small tube or two and sending it off for telomere testing. So blood is a bit more work to get, and testing is obviously going to be a little more expensive because you're doing phlebotomy, etc. However, blood contains many living cells, high-quality DNA, robust literature on telomere biology and correlation with other tissues, and average and shortest telomeres can be provided. So if you're trying to choose, today those are your two main choices, either sputum, saliva, or blood. Okay, the other issues that one has to really think about is the reliability of the test. Are you just going to be spending money to get something that pleases your patient or doesn't please your patient? So is the test accurate within 5 to 10% on repeated samples? That's important. Do the companies have data that they can show you that this work is reproducible? Does the test give you both the percent and the average length of telomeres and the percent of shortest telomeres? While average telomere length may be less useful, since a critical short telomere may be sufficient to initiate cell senescence and disease onsets. And the last part is, without a detailed family history, I'm not sure how useful telomere testing would be, because by tracking people over time, say with annual telomere testing, you will see trends in lifestyle choices. In other words, is there a family history of a certain type of disease? Have these people gained an extraordinary amount of weight? Are these people now exercising when they didn't used to exercise? Things like that. So you can maybe sometimes sort out their telomere testing based on lifestyle choices, and this sort of turns out to be like a tap on your shoulder. That, in other words, if you get a poor result from your telomere test, this is telling you something in your life is probably going wrong, and probably the easiest thing to do at the first level is to change some lifestyle choice that they've been making, maybe take Pilates instead of being stressed out all day or something like that. So, I think one needs to think about having detailed questionnaires when this test is being done. So finally, why would you want to use telomere length testing in your practice? Well, first of all, you can detect inherited telomere disorders in patients, these telomeropathies as I call them, and also you can detect healthy individuals with accelerated biological age. There are people that could be in this room that look really healthy, but it's like a ticking time bomb. They're just waiting for something to happen. Also, by knowing your telomere length, we can enable early intervention lifestyle modifications to reduce stress, inflammation, et cetera, all the things that many of you are interested in here. And finally, in the end, we can provide therapeutic interventions to slow down or reverse telomere loss. Some of you are interested in bone marrow and fat stem cells and things like that. So people with really short telomeres, it may be that their stem cells aren't quite as short as the rest of their cells. So maybe that's a way of determining who is most suited for this. Or we might turn out to take people's own stem cells, transiently elongate their telomeres in ex vivo and give them back their own cells. So it would be a new sort of direction for stem cell therapy. And finally, I just want to show you how modern and active this field is. This is some reports. It's kind of hard to read, but I just put this together on the way up here. In the Science Daily, March 27th, short telomeres are not only a marker of high risk of disease and mortality, but are a likely underlying cause of several age-related diseases, including heart disease and various cancers. This particular team measured over 48,000 individuals and identified 7 genetic variants that are associated with telomere length, and found that these genetic variants also affect various risks. So again, part of your telomere length is genetic, and they now have a test that tell you what causes you to have long and short telomeres, which means in the future we may be able to intervene in some of those processes. Also, in 2013, March, scientists at the Heart Institute in Salt Lake City showed that people with longer telomeres actually live longer. This study's lead researcher, Dr. Carlquist, said of telomere testing, we can already test cholesterol and blood pressure of a patient to see how his treatment is working. But telomere testing could give us a deeper view on how the treatment is working and affecting the body, etc. So if you've got treatments, this may be a thing to consider incorporating into your practice. Again, just some more. Time magazine, scientists at Carnegie Mellon report that short telomeres correlate with susceptibility to common cold infections, of all things. And here's another one, January of this year. I'm not making this stuff up. Researchers from the Spanish National Cancer Research Study showed caloric restriction slows the rate of telomere shortening. So if you have people on special dietary regimens, they may like to know, am I doing any good? Is this diet working, or is your diet better? And by tracking telomeres, you might be able to do that. So anyway, there's more of this, and this will all be on the slides, etc. I just want to end by saying that the analysis of human telomere length has become a central and validated biomarker that reflects biological, not chronological, human aging processes. Accurate commercial tests are now becoming available that will lead to a better understanding of the relationship of telomeres and telomerase to both aging and to cancer. And finally, products currently being touted to promote healthy aging may want to consider incorporating telomere length tests to determine if telomere biology may be the mechanism responsible. And in fact, it will be, as I predict, a new biomarker that's going to become much and much more common. And finally, I would just like to end by saying youth is a gift of nature, but age, as you all know, is a work of art. So thank you very much, and we have a few minutes to answer questions. Yes, absolutely. Questions? Anybody? Yes, sir. Speak up loud. Come up closer. I can't hear you. I'll repeat your question, but come up here closer. I can't hear you. We have a lavalier. Go ahead. Hand mic? No. Is there one up here? No. Go ahead. Here, let me give you my mic. Here comes a hand mic. I just think it'd be easier if everybody could hear your question, sir. We got it? Okay, great. Thanks. Since we've got a few minutes, I'm sure you've got questions.
Is it better, everybody?
Yeah, that's great.
Okay. My name is Dr. Schindel. It seems to me that the telomerase enzyme actually is an anti-apoptic factor, that is an anti-natural cell death factor. The longer chromosomal end you have, the longer your life expectancy may be. And this is a natural kind of a thing that the shortening of the telomeric end could be is a natural process,
sure,
as well as you showed that it could be accelerated by several factors, from nutrition to stress and every other thing.
So what is your question?
You also mentioned about telomerase enhancers, like was it the TA65 or
right.
whatever?
I'm not promoting any.
Okay, I was curious about it because these things are promoted, and I would like to know how does one decide whether it will enhance the activity or rather the presence of carcinogenic events as to compared with,
Okay.
with natural cells or natural individuals.
Okay, so your question, okay, let me see if I can summarize your question so we can give some other people a chance to ask questions. You're saying, how do we know if somebody had a telomerase enhancer, it would be a good thing, basically? That, how do we know that if we elongate telomeres, we're not going to get an increased risk of cancer? And nobody can make that statement right now. In fact, nobody can even make the statement that using anything that's out there today actually has been proven to elongate telomeres in humans, and that it has any correlation with lifespan, enhancing lifespan. It's a correlation right now. And so I would just urge caution until clinical tests are done. So companies that think that they have some natural product that might elongate your telomeres would be well advised to do rigorous telomere testing in a blinded manner, just like it was going to be approved by the FDA. And that way you will know unequivocally with robust tests. You can't just say that my drug elongates telomeres. You need biological evidence. And even in mice and cells, it's not the same as doing a clinical trial on patient samples. So my answer is we can't hype any type of drug. That's just there happens to be one there's only one that's sort of out there right now.
Okay. What we do, and I had a lecture on it yesterday, we have what we call the cancer profile. And these are blood tests which can tell a developing cancer years before you can realize a tumor. So when a person would be applying some of these telomerase enhancers, I think first they should do the cancer profile to see that this individual is tumor-free before it really develops to the point where you can see it with either an MRI or a CT or whatever you have. So these are biochemical blood tests which can be done, and I could make this application quite safe.
Okay. So I just want to correct one thing that you did say in your lecture yesterday. There's not two types of telomerase in the human body. There's only one. The same telomerase that is important in stem cells is also the same telomerase that is in cancer cells. So you do not have two telomerases in the body. And just so you'll know that for the future.
Any other questions?
Questions?
Anybody in the crowd? I guess not.
Must have been perfectly clear. Well, good. So we'll stay a little ahead of schedule. Thank you again for your attention.